Let me start by thanking the organizers for this wonderful opportunity to hear a lot of interesting talks and also to introduce the Taiwan Biobank to the audience. Today, I will be talking about the Taiwan Biobank resources for population health research. There will be a bit of introduction about the Taiwan Biobank, but there will also be a later half where I have more of some interesting observations that we've seen during our exploration into metabolomics using Taiwan Biobank resources. Okay, I will start with a very brief history of the Taiwan Biobank. The Taiwan Biobank was officially established in 2012, so that's almost 10 years ago. And but there was a flurry of pilot studies right before the official establishment. And our mandate was to collect or recruit 200,000 participants by the end of 2023. Now, this is just like a first phase of the Taiwan Biobank project. And as I hope you can see from the slides, I was actually recruiting into the Taiwan Biobank around this point. So that's at the end of 2015. And my original task was to establish a metabolomics program for the Taiwan Biobank. And fortunately, we've been doing that. And right now we have quite some data which I can present to you later on. So the tasks that the Taiwan Biobank conduct are, first, we recruit participants throughout Taiwan. And we are in charge of the actual biospecimen and data collection. These specimens and data are then stored in our central offices or in the National Center for High Performance Computing of Taiwan. So we have liquid nitrogen tanks and freezers, and we also have a small data center and all that for the storage. Of course, you want people to use all these specimens and data, so we are also in charge of dissemination of these samples and data through a formal application process, which takes into account stuff like data security and privacy. Of course, we also conduct our own data generation, mostly in the field of omics profiling, and as you will see in the next few slides. So our aim is to become a vertically integrated biobank, hopefully the largest biobank of Taiwan. We have currently over 40 recruitment or collection stations throughout Taiwan. Excuse me for this being in Chinese, but you can see that there are many stations, and all the personnel within these stations are centrally trained at our central offices here, and then they are sent out to the different stations to do the actual recruitment and collection of the participants. This is to facilitate a single set of recruitment, collection, and laboratory standard operating procedures, just to make sure that the samples we collect are of good quality. And there are more stations in the more highly populated areas. Now, let me show you a bit about the cohort characteristics of the population cohort in the Taiwan Biobank. So like most biobanks, there's a larger composition of females, but the age distribution is about even between the different age groups. Ours is a population-based biobank, so the ethnicity is kind of mixed, although most of them are from the Southern Ming group of the Chinese Han. There are about 10% from the other provinces of Taiwan, of China. And of course, there are also the indigenous peoples of Taiwan, which are more related to the Micronesian or Polynesian populations. We do not carry ICD codes, so our disease statuses are self-reported, but we do have opt-in consent to link our data with various databases in Taiwan, including the National Health Insurance Research Database, which does carry diagnostic codes and treatments. Of course, ours is also a prospective cohort, so we do long-term follow-up. We want to look at how people actually become sick. And currently, most of the data are accessible to researchers worldwide. Okay, this is a list of the releasable materials that we have at this stage. So the target is to have 200,000 participants by the end of next year, and we already have about 180,000 recruited. Now, for each one of them, we get a set of questionnaires for them to fill out, which includes lifestyle information, socioeconomic statuses, and all the general things, much like what the Taiwan Biobank has done. And we conduct physical exams, we get their serum biochem data, and we collect the biospecimens. The biospecimens that we collect include blood plasma, and this is the total number of tubes we have right now, and urine samples. We also extract our own DNA from the Buffy code. And so when they are initially recruited, we call them the baseline participants. They also have their whole genome genotype acquired. And these are just the numbers that are currently available for release. On the other side, because we already had the first follow-up done, about 41,000 of our participants have undergone their first follow-up visit. And we do the same thing all over again for this part. But in addition, we also acquire some medical images with their consent. And these include images of the bone mass density, various types of ultrasound images, and also electrocardiograms. Now, this is the data release process that I talked about. And for international applications, there is a difference from the domestic ones. Everybody has to have its own institutional review board approval. The proposal also goes through the Ethics and Governance Committee of the Taiwan Biobank. And if they both approve, then for international applicants, it goes to the Ministry of Health and Welfare of Taiwan to undergo a data export permit review. And once that's done, then the data can be released. And currently, we already have 3 successful applications from research institutions overseas. Now, let me switch gears and talk a bit about the metabolomic data that we have in the Taiwan Biobank. It is a bit of a history. And the types of data that we acquire here include NMR-based metabolomics for blood plasma. This is mainly conducted in-house because we do have access to some NMR machines here. And also urine plasticizer metabolites, which are obtained through outsourcing. And the outsourcing institution primarily uses LC mass to do that. Now, when we started the project, there was a big question of whether the data would actually be useful, particularly in the NMR type of data, because frankly, metabolomics at that stage was kind of new to virtually in Taiwan. And so we first looked at filtrated plasma samples and did a test where we look at the normal versus pre-diabetic people that we had. Now the pre-diabetic people that we had was using the following definition, that is fasting blood glucose between 100 and 125 milligrams per ml and glycated hemoglobin levels of 6 to 6.4%. This was a very small scale experiment. So we only had 107 pre-diabetic samples and 107 matched controls. And at that time, not knowing better, all the metabolite concentrations were manually fitted. So that took a pretty long time to do. And fortunately, we did sort of recapitulate what has been reported in the literature. So we see that most pre-diabetic subjects, they do have higher levels of these branched chain amino acids. and some of them also had higher levels of acetone, which has also been reported before for other populations. But we did see some interesting, I would say, observations. So we saw that we could detect ethanol and methanol in our samples. And we saw that the control subjects, they usually had a higher level of ethanol or methanol. To this date, we still do not know the reason behind this, but we think that this is actually a good starting point for some research. And this sort of showcases what biobank can do in terms of generating ideas for research in the biomedical field. The second test we did was to test for actual diseases. So we collaborated with several medical doctors where they could get access to colorectal cancer samples. And using our Taiwan Biobank samples as control, which is on this side, we try to see whether there are differences in the metabolomic profile between the two groups. And fortunately, we did see a difference, which is showcased in this principal component analysis plot. You can see that they are fairly well separated. And if we look at the actual data, then we could identify several metabolites or compounds that were different in terms of its concentration in the blood between the two groups. And of course, we also get some unknown signals, just like in mass spec. And generally, we were happy with these test results, but there was a very big problem with this approach because we were using filtrated plasma samples. One of the main problems was that the throughput was not sustainable. I mean, you had to do almost everything by hand. And at that time, we also did not have any automatic sample changers like those at the Taiwan Biobank. So the next thing we tried was to shift to a more raw plasma plus buffer type of simple sample preparation and see what we could get out of it. And this is just to showcase that it is possible to do it and to obtain some useful information, even though usually the number of identified metabolites are much less than using filtrated plasma. And you can link these metabolomic data. This is just the NMR spectrum of raw blood plasma with buffer. And link these with, for example, phenomic observations or genomic observations. So here we look at the linkage between metabolomics, phenomics, and genomics for what we call the tri-high phenotype, which is people with either hyperglycemia, hyperlipidemia, or hypertension. And we use a more chemometric approach, where you do not need to actually identify the metabolites beforehand. You can do that after the analysis. And you can see that there is a clear correlation between these acetone and glycate peaks with people that have a lower tri-high prevalence. And also there is one single nucleotide polymorphism, which seems to be positively correlated with this same phenotype. So this gives you some sort of linkage where you can study further. And because of time constraints, I'm not going to talk more about it. So you can look at the publication directly. I have cited it down here if you are interested. Now we also look at went back again to the colorectal cancer part. And this time, this was actually more like a research project with in collaboration with people who are actually cancer doctors. And what they were interested in was to look at the difference between left side and right-sided colorectal cancer, which for those who do not know, they actually have different etiologies, and the disease progression is also different. Without talking too much about it, suffice to say that we were able to get the spectra from raw blood plasma with buffer, and there were several signals in our NMR spectra that were able to differentiate between left-sided and right-sided cancer if you applied a partial least squares discriminant analysis on it. And this has also been published just this year. But at that time, we came to the realization that even chemometrics is kind of hard for most people. So from the biobank perspective, that was the problem with the barrier of entry, because NMR spectral analysis has a high barrier for most people. It requires quite a bit of training. And chemometric analyses are also difficult for most life science related majors. And what users really want are these neat and nice tables. So in order to get these kind of tables, we changed our protocol, and now we are using automatic liquid handling platforms, customized one to load the samples to have higher reproducibility. And we employ a Brook IVDR metabolomic platform, which gives you automatic reports. There are metabolite differences between different populations, as you can see from this part, and this may be due to these reasons. And the last thing I want to show you, which I will just go very quickly, is that you can see that there are plasticizer metabolites differences between the different populations, and this may be related to lifestyle differences, which I've shown here. And in Taiwan, they're mostly in food, and probably for Americans, which we compare to, they're mostly in the cosmetics part. Okay, with that, I will conclude my talk with this brief summary. I'm not going to read through it, but the most important part is that pan-cohort collaborations are the future, because you can see differences between the different populations. Okay, and I will just go through the acknowledgement very fast. We thank our participants, our staff, the people who gave us the money, and the places where the data were actually acquired. Thank you.